Now, since there is a receptor ligand signaling occurring during the process of activation of T cells, as we just saw, the serial triggering model of T cell activation. Now, one important thing to mind is that if there is an activation, there should also be an inhibition, because a receptor or a biomolecule cannot be activated for a given long time with no half-life. Now, there are two very important molecules that help in the passage of inhibitory signals from the antigen presenting cell to the T lymphocytes. Now, one such molecule, let me talk about this one first. This is the CTLA4, the full form of which is cytotoxic T lymphocyte associated molecule 4. The CTLA4, as you can see, is a disulfide linked homodimeric glycoprotein evolutionarily related to CD28. So they have a relation to CD28. They almost share about 75% sequence homology and have closely resembling structure. Interestingly, CTLA4 binds to B7 ligands with much higher affinity than CD28. If you remember, the signal 2 is actually perceived by the T lymphocytes via the B7 CD28 interaction. So B7 has two receptors on the surface of the lymphocytes. One is the activating receptor CD28 and the other is the inhibitory receptor CTLA4. To be very clear about the fact that CTLA4 has more affinity for B7 than CD28. Now, this poses a problem. When a T lymphocyte is engaging with the antigen presenting cell, if a CTLA4 is already present on the surface, and as I just now said, a CTLA4 molecule has more affinity for B7 than CD28, then automatically, the moment an APC engages with the peptide MHC complexes, the signaling through the CTLA4 molecule will start and automatically the T lymphocyte will be inhibited. So now what happens? So the thing is that evolutionary how this thing has been selected. CD28 is constitutively expressed on most T cells, but CTLA4 is expressed in significant amounts on the T cell surface only after the TCR is triggered and the activation cascade has already started. Because it is thought that the increased affinity of the CTLA4 for the B7 may cause displacement of CD28, disrupting of the co-stimulatory signal, and if the disruption is severe by CTLA4, that will ultimately lead to T cell blocking of the T cell division and the G1S transition. So as you can see, so initially CD28 is constitutively expressed while CTLA4 is not expressed until and unless the signaling cascade has been triggered. So the expression of CTLA4 is tightly controlled. To prevent CTLA4 from acting too early, the majority of CTLA4 molecules in a resting T cell are sequestered in a subcellular Golgi compartment. Within 2 hours of TCR triggering, CTLA4 molecules, I mean to say the Golgi, they are sequestered in the Golgi compartment. So the CTLA molecules becomes reoriented towards the T cell APC interface and continue to accumulate within the vesicles until at least 6 hours after TCR triggering. It is only after 48 to 72 hours of TCR triggering that CTLA4 molecules move out of this compartment, appear on the T cell surface, ready to bind with B7 and damp down T cell activation. So, unless and until a time span of 48 to 72 hours have passed, the CTLA4 are not activated on the T cell surface. But mind it, once they are activated, as I said earlier, they have a higher affinity for B7. So just after their activation on the cell surface, after a lapse of 48 to 72 hours, they engage with the B7, truncation of this signaling is done, and the T cells are then inhibited. So this is how CTLA4 actually brings about inhibition of the, dampening of the, not inhibition, dampening of the T cell activation. Now, coming to the other negative regulatory molecule is the PD1, which is the full name of which is program death 1 ligand. It is a negative regulatory molecule that is expressed on activated T cells. Mind it, it's expressed on activated T cells, B cells, and myeloid cells. Now, the cytoplasmic tail of the PD1 molecule has 2 tyrosine residues that can be phosphorylated, one of which resides in the intrinsic tyrosine inhibitory motifs. The PD1 binds to the 2 ligands. It can bind to 2 ligands, PDL1 and PDL2. Even though in the picture the PD is written as PDL. The important thing is that PD1 expression on a T cell is induced within hours of activation. I'm repeating it once more. PD1 expression on a T cell is induced within hours of activation, while expression of the PD1 ligands like PDL1 and PDL2 on the DC is induced by inflammatory cytokines. So the expression of this particular receptor occurs within an hours of activation of the T cell receptor complex. But the expression of the ligands on the dendritic cells or the antigen presenting cells is not constitutive. They are expressed only in the vicinity of the inflammatory cytokines. When PD1 and PD1 and the TCR are engaged, so when the PD1 and the TCR are both engaged, PD1 transmits an inhibitory signal that shuts down the early steps of TCR signaling pathway. So the moment this engagement is occurring, as I said, the expression of this molecule occurs just within hours of TCR activation. So the moment, if the PD ligand is expressed and this engagement is occurring, the signaling cascade is stopped at the early hours and T cell is prevented from further activation. So, in contrast to the PD1 pathway, as I am repeating, CTLA4 is not expressed on the T cell surface until 1 to 2 days after activation is initiated, giving the adaptive response time to eliminate the threat before T cell activation is damped down. Okay, so this is very important. When we think of this COVID case, now why are that we have an immune system and the virus is infecting our lung cells, we have a good adaptive immunity against the viral system, and yet why is our immune system failing? So in case of COVID now, time and again, you have heard that there are production of a huge amount of inflammatory cytokines in the vicinity. And as we just now read that inflammatory cytokines results in the expression of the PD ligand on the dendritic cells. So the moment these ligands are expressed on the dendritic cells, what they do? They are now actually inhibiting the already activated cytotoxic cells or the helper cells, bringing about a down regulation of the immune response. So this structure is of a T helper cell interacting with an antigen presenting cell. So as you studied today itself, that the helper cell acts as an important pivot between the B lymphocyte and the T cytotoxic. So if there are a huge amount of inflammatory cells in the lungs, as inflammatory cytokines in the lungs, the dendritic cells in the lungs, it's a mucosal tissue, will be then exhibiting this PD ligand, as a result of which the helper cells in that area become immediately down-regulated because of the PDL, PD1 ligand signaling cascade that starts. So remember that this is also a strategy in which our body suppresses inflammation, because a pro-inflammatory activity going on for a long time in a particular area is not beneficial, because inflammatory cascades tend to gradually kill the innocent bystander cells. So their evolution has given us a backup strategy to the cells, that if there is a heightened inflammation, gradually start turning off the cells that are bringing in or recruiting more inflammatory cytokines or recruiting more immune cells. So in case of an extensive inflammatory reaction, it often happens that the dendritic cells gradually turn off the T helper cells to bring the tissue setup into a normal condition. So that's a normal evolutionary phenomenon. Now, as in case of COVID, we saw that a huge amount of inflammation is occurring within the lung tissues. So this phenomena is also occurring there, where the immune regulator, the T helper cells are becoming down regulated, so that the adaptive immunity as well as the humoral immunity, both of the immunity are taking a back seat. So for today's class, this will be our last picture, and this is the time after TCR engagement and post-stimulation. Now, as you can see, there are certain genes have been indicated, and you can see these are the genes that are activated within 15 to 60 minutes, hours within 2 to 20 hours, and days within 3 to 14 days. So you can see the life of an effector cell usually stays for 3 to 4 days. Once we get the progeny, and we are getting the generation of effector cell at the end of 7 days. So it's it's a 0 minute to 7 day reaction, almost. It's 5 to 7 days reaction that gives us effector cell generation, and the effector cell generation population stays for 2 to 3 days, maximum 4 days, and then die off. Now, within the first few minutes, the immediate genes are expressed. These are the different genes, and within the next 24 hours, we get the expression of the early genes, and finally, the expression of the late genes comes from 2 to 3 days. Okay, so this is how the TCR engagement, gene expresses once the T cell receptor is triggered. আচ্ছা করে আমি আজকে sorry